Assalamualaikum warahmatullahi wabarakatuh, and good morning, UMHealth families. We are back for our weekly breakfast at UMHealth on air every Wednesday, organized by Viacom FOM. Thank you everyone for tuning in this morning. I am Intan Zulkifli from Department of Anatomy FOM. I'll be the moderator for the first session. A little housekeeping, in the interest of time, kindly please submit all your questions in the chat box, and I'll read them to the presenter once the session ends. Okay, ladies and gentlemen, as you can see today, we have this darling lady from Anatomy Department, Associate Professor Dr. Rosie Pamela David. We have known each other for 10 years now, and I've always known her as an excellent friend, amazing lecturer, but more importantly, a very dedicated neuroscientist. Today, she's going to share with us the findings from her work. Her presentation today is entitled The Neuroscience of Learning and Memory, Diet-Imposed Brain Plasticity. The floor is yours, Prof.
Thank you so much, Dr. Intan. I almost forgot what I have to say. Okay, let me just share my screen first. All right, thank you so much again for everyone who is here attending this talk. So this is the title page of my talk today. And as Dr. Intan has introduced, the neuroscience of learning and memory and diet-imposed brain plasticity. So neuroscience is made up of many branches here. And you can see that, hold on, I'm not getting the... Yeah, so neuroscience broadly means not only the studies of a normal function, but also the neuroscientists will focus on the brain to study its behavior, how it's developing, and what are the structures that make up this complex organ in our system. So there are many branches to neuroscience. And we can see here, cognitive neuroscience is what I'm very interested in, and it studies the higher cognitive functions that exist in humans and their underlying neural bases. Behavioral neuroscience is also something that I'm doing in my current work, and it looks at the biological basis of behavior and looking at how brain affects our behavior. So there are many different branches here, and it is a diverse amount of knowledge to begin when doing research in the neuroscience. So next will be, I'm going to go into some basics here, and in my talk you'll notice it's not too technical because we have both the medical and also the scientist group in our list of attendees, and also so therefore I'm going to go into some basics. So let's begin with looking at this diagram here, and we can see two neurons are placed here, and neurons are the fundamental building blocks of our nervous system. And communication is through the neurotransmitters which signal across the synapses. So synaptogenesis is actually the formation of new synapses or molding of existing synapses, which is particularly evident during early childhood. But it also continues into the adult brain. So here you can see many cytoplasmic processes that are entering the cell body. These are the dendrites, and the axon actually brings out the electrical impulses, and you can see the yellow arrows showing the direction of the electrical impulses being transmitted out. Learning, this is the process of acquiring new knowledge, skills, behaviors, attitudes based on the experiences and also our interactions with the environment. So synapses become more efficient after repeated activation. So this long-term synaptic plasticity was first reported in 1973, and studying it in a pathway of a rabbit's hippocampus, researchers discovered that rapidly and repeatedly activating the synapses made them stronger, and this was called the long-term potentiation. The reverse phenomenon exists in which synapses become weaker over extended periods of time, and this is called the long-term depression. So long-term synaptic plasticity forms the model for memory storage. In 1949, Donald Hebb actually proposed a pioneering theory in the field of synaptic plasticity and memory. This theory proposed that neurons which fire together are wired together. The neuronal connections that fire together are strengthened, and so those neurons are activated together during memory recall. So memory is actually the process of taking in information from the outside world. This is called the encoding, storing it in our brain. This is storage, and later recalling the information to answer a task or solve a problem or even make a decision or elicit other responses. So these are the three basic stages in memory processes. So there are many types of memory. Okay, we can see here, but the details will not be discussed here. Perhaps the distinction between working memory and also long-term memory is fundamental to understand how cognitive sciences can be applied to teaching and learning. So working memory is our processing unit where we information to solve has very limited capacity. It is like a shoebox, offers limited space and cannot contain too much information. Example, when you're trying to remember a phone number long enough to dial it, you are using your working memory, and soon after you have done it, you will probably forget it. Okay, so you're just using that working memory to retain and use the digits in sequence. So that is one example of short-term memory. And even when we are navigating in unfamiliar place, say you need to attend a webinar in UPM and you hold a mental map of your surrounding, how to waste there, and that is all involved in the working memory as you drive and make decisions. Whereas the long-term memory, this is our storage box. It can store unlimited information for an entire lifetime. And this example is, of course, like remembering birthdays and anniversaries, your favorite nursery rhymes. So this all comes under long-term memory. We can think of long-term memory as a Mary Poppins bag. You can fit as much as you want in there and you will be still able to go around or even fly around. Coming into a bit of neuroanatomy, which is my background, hippocampus is actually a complex brain structure embedded into temporal lobe, deep into the temporal lobe, and it plays a major role in learning and memory, including consolidation of new memories. Okay, it's an important part of the limbic system that's also involved in regulating motivation, spatial orientation, and navigation. And see, there are different parts of the hippocampus that are usually studied during researching into the learning and memory of the hippocampus. And in this dissected picture, you can see how a hippocampus looks like deep in the temporal lobe. Now, brain neuroplasticity or brain plasticity is a remarkable ability of the brain to adapt and rewire in response to intrinsic and extrinsic stimuli by reorganizing its structure, function, and also the connection. So understanding the brain plasticity offers insights into learning, recovery, and adapting our ever-changing world. So this image here shows how neurons in our brain form new connections at an astounding rate of 700 per second in the first years of life as the baby learns to look and hear and start to make movements. So this is happening then. And after this period of rapid proliferation, there is also a time to reduce the connections through a process called pruning. So there is a time for proliferation and there is a time for pruning as well. So brain circuits become more efficient and sensory pathways like those for basic vision and hearing are the first to develop, followed by other cognitive functions. So these connections, as you can see, how the cytoplasmic processes have really diversified their connections. These connections are the building blocks of the child's future. So how diet affects the brain plasticity. So here we can see a typical fast food or diet or even junk food that we easily grab on a lazy or tired day, or even when we simply decide to dine out with our children who are in that teenage stage. This is usually what their favorite outlet is. And there are many effects of this bad diet on learning and memory, and those include the inflammation and also cognitive impairment, because these diets contain high trans fat that promote chronic inflammation. And we can see on this side the healthy diet, which is made up of a balanced mixture of vegetables and fish, nuts, and so on. So these are nutrient-rich diet which contain nutrients like omega-3 fatty acids and antioxidants. So this well-based diet is connected to a healthy brain. They also, in aging brains, they also are related to age-related cognitive decline. So these are brain boosters for aging as well. Obesity, this is defined as an imbalance between energy intake and expenditure. So the BMI exceeds more than 30 kg per meter square. And according to the World Health Organization, Malaysia is the fattest country with almost half of the population being overweight. Okay, nothing to be proud about, but something that we should be working into. Okay, why are we so fat? It's not just because of eating junk food, but because of eating imbalanced food where there is lack of vegetables and fruits. With the rising trend of obesity, the incidence of gestational obesity is also on the rise. Okay, a study among globally pregnant women estimated about 38.9 million to be overweight and obese. In Malaysia, reports from the National Health and Morbidity Survey observed a high prevalence of obese pregnant women, and the commonly reported complications in pregnancy including the miscarriage, gestational diabetes, and also preeclampsia and stillbirth. Another concern of gestational obesity is the health risk imposed on infants that they carry. And these infants, there are a likelihood for them to develop the non-communicable diseases into adulthood, and offspring neurodevelopmental outcomes are also affected. So that's a bit of a background before we delve into what my research has been on with regards to this topic. So basically, one of my research interests is it touches on the effects of obesity or unhealthy diet, namely high-fat diet on the nervous system. And we explore how functional or medicinal mushrooms are able to mitigate the ill effects of high-fat diet on the brain with regards to learning and memory. So a high-fat diet, particularly one that is high in saturated fat and trans fat, can give rise to a number of the following effects, such as the cognitive impairment, which leads to difficulties in concentration, problem solving, and also memory recall. Neuroinflammation is there. When triggered, it can contribute to various neurodegenerative diseases. Then we also see impaired neuroplasticity in these individuals, because we know that synaptic plasticity is essential for learning and memory establishment. And so forming and strengthening new connections are hindered when this kind of diet is practiced. Also, there will be interruption in the blood-brain barrier. So these are some of the ill effects of high-fat diet. And we in our research use a variety of mushrooms to see the alleviation of the ill effects in the rodent model. So we use a variety of mushrooms. This is actually the Pleurotus pulmonarius, Cendawan tiram kelabu, and we are particularly interested in the beta glucan from this mushroom for our study. So for your information, beta glucan is a type of soluble fiber which is a naturally occurring polysaccharide widely present in many other foods Oats is one good example, and even like barley and so on. So it's associated with numerous health benefits such as weight gain prevention via lipolysis, like hyperlipidemia prevention, and it also exerted antioxidative properties. So one of my students, Miss Nur Atira, she worked on the effects of beta-glucan-rich extract of this mushroom, and she assessed the object recognition memory, and she also looked into the hippocampal morphology in mice that is fed with high-fat diet. So beta-glucan, you may wonder why from this mushroom we are using it and not from other sources, because the structure is different between different resources. So in this study, we actually evaluated the effects of this beta-glucan in female ICR mice with that has been fed for 3 months and to develop obesity, and the female mice were subjected to few neurobehavioral analysis, but upon mating and also pregnancy, there were offsprings there, and these offsprings were also tested for their neurochemical structures, and also histopathological and immunohistochemistry was carried out in their the hippocampus as well, to see the effects of high-fat diet and whether the beta-glucan was able to alleviate the ill effects. This is one of the tests done, novel object recognition test, and this is to assess the object recognition memory. I'm supposed to play the video, but I'm not seeing it. Okay, so you can see while I talk. So in brief, during the acquisition period, the mice is exposed to 2 identical objects, but after 1 hour, we test the short-term memory, and here the mice is presented with a familiar and also a novel object, and after 24 hours the long-term memory can be tested, whereby the mice is presented with a familiar and another novel object. So mice with good memory will explore the novel object, and increased exploration suggests that the animal recognizes the change and has a functioning recognition memory. Okay, so these are, I'm not going to go into the details of every results in the interest of time, but analysis of the results on the object recognition memory of maternal dams showed that high-fat diet group indicated a slight deficit in short-term object recognition memory. This is 1 hour, and BGPP supplementation, the beta-glucan from Pleurotus pulmonaris supplementation, significantly improved the short-term object recognition memory. However, there was no significant difference observed after 24 hours. As illustrated in the figure, we can see that there is the effects of BGPP supplementation on the maternal hippocampus showed also significant amelioration, or in the significantly ameliorated the loss of neurons, and also reduced the number of EBA1 positive microglia in the high-fat diet fed mice hippocampus. This is in the offspring. At day 14, we notice similar positive results, but at postnatal day 28, the results were not significant. In another study, we looked at the effects of this mushroom, the Hericium erinaceus mushroom, and we looked at the effects on the HT22 hippocampal neurons and also the BV2 microglia. And in this study also, I'm just going to concentrate on neuroprotective mechanism and also the neurobehavioral aspects of the study. So in this study also, we saw that the Hericium mushroom protected the hippocampal neurons against hydrogen peroxide-induced oxidative neurotoxicity, and they improved the antioxidant activities in these neurons as well. And this slide shows the neuroprotective mechanism associated. We can see there is a significant reduction in the ROS level, and also the catalase and GSH activities are enhanced by the mushroom. So this is the Hers nuclear apoptotic staining. This also shows that there is reduction of nuclear apoptosis in those treated animals. Treated cells. And this is to the Y-maze spontaneous alternation test to assess the spatial working memory. We can see the mice is actually exploring the different arms of the Y-maze, and this assesses spatial working memory. So spatial working memory, mice with a good memory will have a high spontaneous alternation, but unfortunately the results were non-significant. So basically, in vitro studies showed that the mushroom protected the hippocampal neurons against hydrogen peroxide-induced oxidative neurotoxicity, and the live animal studies showed that the hericium attenuated anxiety behavior and significantly improved the object recognition memory for short term. Finally, mounting evidence has demonstrated the deleterious effects of high-fat diet on cognition. And the beta-glucan supplementation in our study and even the mushrooms, like elsewhere, has shown immense neuroprotective effects. Many studies are researching into the involvement of gut-brain axis. These are studies not done by me, other studies. So many studies are researching into the involvement of gut-brain axis, suggesting a vital role of the gut microbiome. One thing that we the mechanism that plays a role on how maternal obesity influences the neurodevelopment of learning and memory in the offsprings, and see if beta-glucan from these tasty, yummy, meat-like textured mushrooms can whether they are able to adverse the developmental to prevent the adverse developmental outcome. So these are just some of the references. This study was supported by the Research University grant, the RU grant, and students who worked on the two projects I showed were Naufal and also Nur Atira, and most of the work was co-supervised with Prof. Vicky from Science faculty, Prof. Murali, a fellow member in our department, and Dr. Panchawe from the pharmacy side. With that, I thank all of you for your kind attention.
Thank you very much, Prof. Rosie Pamela. It was a very good talk. Prof. Tukusara commended your work, Prof. But she wanted to know a few things. Firstly, if we want to eat these mushrooms to help our brains, what is the best way to cook them? Or does it matter? Or should we take the beta glucan supplementation instead?
Okay, so the best way to cook mushrooms is basically don't overcook them. Okay, many of them prefer to eat them in a soupy way of preparation. Okay, sometimes we fry them. It is more tasty when it's fried, but then we are again using oil, and we are again dumping in more oil and unhealthy stuff into us. So although it doesn't taste as excellent, but more of a soup base will be better for the intake of these mushrooms. Beta-glucan wise, it's one of the polysaccharides contained inside, and I'm not very sure whether they are isolated and sold as supplements yet, but yeah, you can consume it as a whole. That's much better.
Thank you. One more question. What was the high-fat diet that the mice eat?
The high-fat diet mice was gave a concoction of different nutrients, but the fat itself was almost about 60 to 70% of that diet. And we got this diet from a German company called Altromin that specializes supplying diets for these animals. And you can actually mention how much percentage of fat you want, even if you want it customized.
Okay, we still have a few minutes. There are two more questions. The other one is, would drugs with the correct dose of beta glucan be a good substitute for consistent dose for our patients?
Okay, so beta glucan to be incorporated and given to the patients, we have not done clinical trials on that. So I wouldn't comment much on that without having any clinical trials done. Okay, that is a future, something that we can look at in the future with more funding.
And last one, hi, good morning, doctor. How much of the mushroom is needed to get the desired effect? Is the effect dose dependent?
In rodents, yes, we did a pilot study with showing like for for Pleurotus pulmonaris, we did a toxicity study and we did different dosages. And so we found that even at the highest dose that we tried out, about 500 mg per kg of the rat, we didn't find any toxic effects, but we didn't do further than that. The LD50 toxicity studies was done for some of the mushroom. We didn't find any lethality in it, but yeah, for humans, we haven't done any work on this. But I must say there is a lot of these mushroom products being sold in the market. Okay, we can go to the supplement store and a lot of them. And I myself am taking one kind of tiger milk mushroom to help me with lung issues. It does help me a bit. It feels better, you know. So they sell it in the pharmacy and it does help. So there's a lot of different varieties, but be cautious in whatever you take. Review it beforehand. Yeah.
I think this is the last one again from Tengku Sarah. I thought that fat is no longer the bad element in diets, replaced by sugar. What is your comment on that?
Come again?
As she said, I thought that fat is no longer the bad element in diets, replaced by sugar. What is your comment on that?
Of course, there are the good fats that we are encouraged to eat. So again, but sugar is not, from what we hear these days, people have gone into even omitting sugar from their coffee intake. So yes, sugar is almost like a no-no. You have to be very careful with the intake of sugar. But there are good fats. You just need to know which ones they are and take them in proportion.
Yeah, thank you very much, Prof. I'll pass the moderation to Dr. Chai Hua Chia for the next presenter.
Thank you so much, everyone.